All right, as we wait for the last few folks to join us, a few housekeeping notes. We envision this as an opportunity for a low-key, informal educational presentation. We're highlighting the features of our instrument and software, but the focus will be on how to make meaningful contractility measurements from iPSC-derived cardiomyocytes, as well as how to analyze and interpret that data. Our goal is that you walk away from this with an understanding of what it takes to make the measurements properly and what the analysis tells us. With that said, this is a standard Zoom meeting. It's participatory, and we're hoping that you will feel comfortable asking questions. But we've got a good number of people that have expressed interest and have already joined us today. So things will get exceedingly chaotic if everyone asks questions at the same time. So please keep yourself muted when you're not asking questions, so that we don't hear your dog barking or your lab mates screaming obscenities at one another. Okay, enough of that. Let's get started by introducing my colleague, Emmy Manders. Emmy is our Manager of Customer Satisfaction and one of our internal resources for designing, testing, and validating our products. She's been working together with the VU in Amsterdam on a number of ongoing projects as well. We have a good relationship with the folks at the VU, and they've been kind enough to provide the iPSC cardiomyocytes today. Emmy's going to briefly discuss the setup and then tell us what we'll be doing today. Again, the focus is on education, so please feel free to ask questions if there are any particulars that you'd like to know more about. And just as an FYI, I'll be uploading the recording of today's presentation to our YouTube channel and embedding it on our website. We'll also put together some application notes and upload those to the website. When they're up, I'll send an email around to let you know that they're available. All right, Emmy, you have the floor.
Hi, everyone. Thanks for the introduction. So indeed, today I will show you how you can do measurements on iPSCs. I'll first show you the hardware that we use and the dishes that we have. And then after that, I will switch to my screen where you can actually see the actual recording of contractility. So in my screen now, you can see the top of the MultiCell microscope, but the lid is closed, so you can't really see that much. But if I open it up, you can see that I have a plate in here, and I have a diamond plate in here so that I can actually also fit well dishes in there. So in this case, I have a 12 well lid, 12 well plate in here, as well as an EBD 24 well plate. It's hard to see because it's also black, but it's in there. So unfortunately, in the EBD dishes, our cells were not contracting that great. So we had as an alternative the 12 well plate. This is not a standard plate that is used for measurements, and we don't also supply anything to pace in a 12 well plate. So we have to rely on spontaneous contractions in these plates. But usually if you do the 24 well plates, then we do have a 6 and 24 well pacing solution, which you can just fit in there, so you can pace, or you can just at least set 6 wells up at the same time. And then you can pace the cells, or you can just look at spontaneous beating cells. So that's the microscope. Let me see how I can actually take off the, oh, sorry, not that used to this yet. Okay. Can't seem to get the camera off. So I think the main thing is that we have a myopacer to stimulate the cells, and we have the excitation light that's coming in from the back. So there's a Kohler illuminator at the back of the microscope that really focuses the excitation light to only a very small part, oops, of the cells, and then the emission will go through a couple of dichroic cubes inside the microscope and also on the outside of the microscope. So here you can see one dichroic cube that will send the emission to the PMT, and the rest of the light to the camera. And because the excitation is switched, so it's dual excitation measurement usually for Fura measurements, which switches rapidly between the 340 and the 380 LEDs. You get the ratio of those two to get your dual calcium measurements. I think that's the main part of the hardware. I'm not sure if there's anything unclear or that I maybe have missed. If not, then I'll switch to my computer screen. Let me share the correct screen. So this is the Iron Wizard. Are you seeing it? No.
Yep, we're seeing it.
Oh, okay, sorry. Yeah, so this is the Iron Wizard main screen. I've set it up for now to only measure on contractility on these iPSCs because the 12-well plate is a plastic bottom plate, which enables us to use it for calcium loading or calcium measurements. So we can only measure contractility in these well plates. So I set it up to only measure contractility, and you already see a little bit of a preview of the cells contracting. And then if I open the cell finder window, we move to the MultiCell view, in which you can see on the left a 9-panel view of the complete dish, and on the right is the live view. So you see the cells or the monolayer beating. So this is a monolayer of iPSCs, so not single cells contracting or embryonic bodies, but it's more or less a monolayer. And if I switch focus, yeah, you can see parts of it moving more or less. So that way we can navigate throughout the dish by just clicking through it, looking for regions that are of interest. And if I want to start a measurement, I simply press start. And if I switch to the multi-cell window, we haven't measured anything yet. That starts with an add measurement. So the region that we are measuring is the orange box. And if you would do calcium measurements, we excite only the region that is within the blue circle. So that way, if you measure both calcium and contractility, you measure in the same region exactly. So you don't excite the complete dish and you don't get the emission of the complete dish, you just get it from a very small region where you also measure your contractility. So the method we use to measure contractility is either pixel intensity or pixel correlation. I've set up the experiment to show both. So we can discuss when we do a couple of measurements, what the differences is between those two methods and why we prefer one, but both are incorporated. So in this case, I manually measure locations of interest. So I just click around in a dish and see where there's a lot of contraction and also where it more or less contracts to the center. So you don't see movement in one direction, so only going vertical or only going horizontal, but you see movement towards one point more or less. That's at least the regions we think are most interesting because that's the region that is actually contracting. So to measure, I simply press add measurement, and then we can already see the traces appear here. Let me make this screen a bit smaller so you can see better. So the top is showing pixel intensity, and I'll switch this one to pixel correlation, and then I'll change the view. So what we see here is two measurements of a contraction in that region that I just, well, what you can also still see here in the bottom. So what our method does, it finds first a reference frame, and the reference frame is taken in diastole. So what we do, we collect a couple of images throughout a second roughly, and then we determine what is the reference frame based on the changes within those that image stream. And when it's more or less stable, we consider that as the diastolic part of the contraction, and we take the reference frame. And then from that reference frame, we for pixel intensity, we look at each individual pixel, and we look at the changes in pixel intensity for that pixel throughout the measurements, and we measure at 250 frames per second, so a very high rate, we calculate the changes in pixel intensity. We sum that up and divide that by the number of pixels, and that's how we end up with a number of pixel intensity changes. So then if it's similar to the reference frame, pixel intensity is roughly 0. And if maximal contraction, so then most of the pixels have moved or changed in intensity, it's at maximum. So the peak of contraction, which is then, in this case, around 17. For pixel correlation, it's roughly the same method. So we use the same reference frame, but instead of looking at the differences in pixel intensity, we look at the correlation of each pixel. So if you look at one pixel and you look at the pixel in the next frame, you can calculate the correlation between those two pixels. And if the correlation is about 1, then they're exactly the same. So there has not been any change. And if they're completely different, then the pixel correlation number is lower than 1. And then all that information is summed up for each pixel and also taking the average. So in that way, you end up with at baseline, it's roughly, so when in diastole, it's roughly 1. So there's hardly any change in the with respect to the reference frame. And then during contraction, more of the pixels are changing, which results it going well towards 0, at least. Yeah, it's decreasing, and then there's a maximum decrease. So what the advantages of pixel correlation is that it's always between 0 and 1, which makes it easier if you have a couple of measurements. You will see that the pixel intensity number can vary quite a lot, while pixel correlation is more stable. So let me just go forward with just measuring a couple of regions. Yeah, and if there are questions in between, I can try to answer them.
So Eugen asked a question through the chat, which I responded to, but just in case anybody missed it, Eugen asked if you're measuring from a group of cells, and the answer to that is yes. You can adjust the ROI to restrict the number of cells, so you can make that ROI smaller. But unless you have isolated cells, you're always capturing everything within the region of interest.
Yes, maybe also interesting to add is that for the excitation region, we can go slightly smaller than the current blue circle, which is roughly 100 micrometers in diameter now. But physically we can't go that much smaller. So if you want to make a clear comparison of contraction contractility, we try to have those the same size. Of course, if you only measure contractility, you can make the ROI as small as you like. Another remark is that we have tried to also measure in single cells, but in our hands, at least so far, as a single cell, the iPSCs have not been that happy. So they don't contract well, or they hardly contract at all. But that's just the experience we have in the lab. So we haven't been able to measure that much on single iPSCs. But maybe if people have better experience with culturing, then it is still possible. So this method of pixel intensity or pixel correlation works on anything that moves, basically. So either it's a very small region that is contracting, or it's a big region. It captures all.
So Kevin asked another question. Have we tried this on primary adult cardiomyocytes? And I don't know the answer to that. I don't know if you've ever tried this, Emmy, but we do have other algorithms that are dedicated for primary adults, including a cell length and a sarcomere length algorithm. So normally when we're using primary adults, we use the other algorithms to capture contractility in those cells.
Yes, but we also did make the comparison in the adult cardiomyocytes. So you still capture the dynamics of the contraction. So the speed of contractility, the speed of relaxation. But one of the downside of the current method of this pixel intensity is that the absolute change, so the peak height, does not really give you information. So if you're For adult cardiomyocytes, you can look at the percentage of shortening by looking at the changes in sarcomere length. And with this method, you wouldn't be able to use that value. So that's also something to take into consideration of how you measure in adult cardiomyocytes. But for example, if you have adult cardiomyocytes that have a poor striation pattern and you can't use the sarcomere length method, then this could be a good alternative. So in the meantime, I'm just clicking away and finding regions of contraction. Well, you can see I changed the focus. I do that with shortcut keys, but I can also just press these buttons here. So just to have them roughly in focus. And after I've done a couple of measurements, I was planning to do about 20. I will add ISO to this dish, isoproterenol. And then, yeah, we can measure the exact same regions again and see their response to ISO.
Another question just came in. I mean, does the difference in pixel intensity, peak to trough, correlate to percent shortening?
No, not really, because the height of the peak and also for pixel correlation, that's the same. Roughly, it really depends on the number of pixels that are moving. So it's more an indication of how many pixels are moving than it is about how strongly they are contracting. So for example, maybe I can find a region that is only very small region that is actual contracting. You will see that the peak height will be lower. Let me see if I can find a, I think this one should have a smaller peak. Yeah. So clearly, so for this region, there's only a very small region of the total region of interest that is actually contracting. And as we take the average of all the pixels in the ROI for pixel intensity and pixel correlation, you can see that the peak height is a lot smaller. But this doesn't imply that the cells or the region that is contracting is not contracting strongly. So the peak height only gives more information about how many pixels are actually contracting than how strong they are contracting.
Another question from Renee. Is it possible to measure cell length by edge detection and contractility by CytoMotion within one acquisition file? And is it possible to calculate both by CytoSolver?
Yes, you can add different methods on top of each other. So I can do sarcomere length, edge length, CytoMotion all at the same time. And CytoSolver should also be able to handle that. It just takes every trace separately and analyzes that. So yes, I think that should be possible. Using edge length in these cells might be tricky, but I guess that depends on the constructs. If you more have embryonic bodies, then that might work. But yeah, anything can be topped on top of each other. So when I'm clicking away, what I'm trying to avoid is to have within my ROI a region that is contracting in a different rhythm or in a different state. So because that would induce additional peaks in your trace. So I think this one might be an example of that. Let me see if I am correct. So there's a couple, so I see contraction here, but I also see contraction more on the top, which do not seem to be completely synced. What you can see happening is that you get, let me zoom in a little bit more, is that you get these extra bumps in your trace. So those are the regions we try to avoid. That does require some experience, I guess. If you do this more often, you get a better feeling of when those additional bumps might appear. But those are the things that I look for when I try to measure. I want to measure one single contraction, so something that is contracting in a similar rhythm and synchronously, so that you don't get additional beats, which will become tricky to analyze.
A couple more questions came in. Can CytoSolver automatically identify, measure, and average unpaced contractions, which I think you're going to show? For example, your cells currently are not paced. Will CytoSolver be able to identify where to start analyzing, and can it identify beat-to-beat variability?
Yes. So the short answer is yes, and I'll show in a minute. So I'll finish up these measurements, and then we do the ISO experiment, and after that I will switch to the analysis side of these measurements and show you how and what we can get from these measurements, also if they're unpaced. So if you wait a second, then I'm happy to show that.
And one more, how do you compare data if you have different cell density?
That's a tricky question. I don't have a clear answer on that, honestly. So the cell density varies also throughout the dish. So we don't control that. We only control the region that we're measuring. So I always use the same region of interest if we want to compare measurements. So this roughly is a region of 100 micrometers by 100 micrometers. But yeah, it also, I guess if you have a very standardized protocol, how you seed the cells, how you differentiate them, it goes for the whole process. If you do that in a very strict manner, we have seen from the few people that they have been able to have quite a low variability between differentiation cycles and between wells. So I hope that more or less answers the question.
And one more question. If the amplitude is artificial, which calculated parameters are in relation to contraction? And then Renee goes on to say, I guess the different times we can use, but velocities?
Yes, indeed. So the focus for these type of measurements is really on the kinetics. So how fast they contract and how fast they relax. So if we move to the CytoSolver, the output is the same as it is for adult cardiomyocytes, but there are a couple of parameters that are of less use in iPSCs and some are more of interest. So especially indeed the time to peak values, which can either be time to peak 50 or full time to peak, and the time to baseline values can be of interest, and the peak width, and also the time to maximal velocity. But again, velocity itself, so return velocity and departure velocity, do also depend on the height of the trace. So those are also not a reliable measurement, as they depend on the number of pixels that are contracting. But the time of maximum velocity can be used.
And one more, is the position of the recording stored so you can use it again, for example, after ISO?
Yes, it does. So what we see on the top left here is are these green little dots. That each green little dot is a region that I measured. So if I switch to the free view mode, let me just make the screen a little bit bigger so you can actually see it. I can just easily switch now between the different locations. So the MultiCell microscope has an objective that can move in an XY position, and that information is all stored with each measurement. For each measurement, you can also save the image of the measurement. So if you're interested in what the cells actually look like when I measured them, and did they change during measurements? So if, for example, you add a compound and the whole structure changes because, well, the cells didn't really like the compound, then you could also see that afterwards in the images that are collected during all these measurements. So now I measured 20 locations. I will press complete, and I will add the ISO to the dish. So if I just quickly stop sharing my screen, I think you should see how I do that. Oh, wait, stop share. So with iPSCs, their response to ISO is a lot less compared to adult cardiomyocytes. So for this, I'm using a concentration of 1 micromolar of ISO. So that's really quite high compared to adult, where we usually do 30 nanomolars. For iPSCs, we have also had good measurements with 500 nanomolars, but below that, you hardly see a response to ISO. So what I do is I have 100 microliters of thyroid, which is the medium that we're using. And I just added a very small amount of my stock solution of thyroid, and I just drip that inside the dish, and I can see a response quite quickly. I'll try to be out of the way. So I try not to touch anything else to make sure that we are still in the same locations. If you have a pacing lid in there, that sometimes becomes a bit more tricky. And especially in these manual 24-well plates, you really have to be careful not to touch the plate because then you get a slight shift and you're not measuring in the same location again. So let me switch to my screen again.
More questions, Emmy?
Yeah.
Oh, it went away. Hold on one sec. Okay, how old are the cardiomyocytes? That's Francisca's question. And then Eugen asks, can the machine select the regions automatically for the first round before the introduction of ISO?
Yes, so the first question regarding the age of these cells, these are roughly 30 days old. So we measure usually between 25 and 40 days. That's the protocol that we use here. We also have younger cells. It really depends on your experiment. So we also had measurements around, I think, 16 days, because then afterwards, those cells were used also for creating EHTs. So they had to be harvested after that. You could also do prolonged experiments if you want to just look at how the cells start behaving when they, well, get older. So anything is possible, but yeah, the basic is for us is 30 days. And these cells were plated on Monday. So usually we plate them 4 days before we do the actual experiment. So I think we can already see that there's some response to ISO. I think the beat frequency has gone up a little bit. I think we can wait another minute or so to let them well, take up the ISO and develop the response.
One more question, Emmy. Can the machine select the regions automatically for the first round before ISO?
Right, that was the other question, I forgot. Good to mention it again. Yes, so we have developed a method to find these centers of contraction, but we are currently still validating it and giving, building up experience on what are the best settings to use, how you can adjust those settings, how to make them work for different constructs. So it is implemented already, and I'm happy to show it once I've done this round to see how it performs. I think we can still do some small optimizations on it, but especially if you have a monolayer that is really contracting almost everywhere, then it responds quite well. If you have very small regions that are contracting, you might be faster by hand. But I'm happy to show that after this round. So if I want to measure these same locations again, because everything is stored, I can just press automatic measure and it will just quickly go through all the regions. I will switch to the other screen so you can see how these measurements look. So we measure these regions all for 5 seconds. You can set up the experiment for longer measurements, especially if you have lower beating frequency. So if you have a very slow beating rate, it might be useful to measure for 10 or 20 seconds. Or if you're especially interested in changes in beat frequency or beat frequency variance, then you want a certain number of beats to get a best readout for that information. So it really depends on the, well, what you're interested in, how you would set up the experiment. But this is just the easiest, well, shortest time, I would say, to show how it's contracting and if it's a stable contraction or if there's irregularities. So this region is definitely different from the other ones.
Another question came in. Have you noticed any difference in response to ISO in cells 16 days old versus 30 days?
I don't have that information on the top of my head. I think we have that data, but I don't know the result of that. Sorry. I guess it also depends on how you mature them. So some people do metabolic selection, some people don't for numerous reasons. So if they're more matured, then their response to ISO will most likely be better. So it really depends on the maturation. So I would say normally if they're younger, then the response would be less, but I don't have the data by hand or know for sure that it's also what we have found so far.
One more question. Do you need the MultiCell system for these recordings, or would it also be possible with the MultiCell Lite?
Yes, so we also have the MultiCell Lite system. So then you have a top stage that is moving instead of the objective. Then you can still do the same measurements. The software is practically the same. It's only the movements that are a bit slower. And with a standard microscope, you have to consider that it's not temperature controlled. So you have to find another way to keep the temperature up, or you have to be very fast, I guess, because iPSCs don't tend to like it if it gets colder. Another option is the stem cell light option. So then you only acquire a camera and our software, and you can measure also on iPSCs on any regular microscope you have that can have a camera attached to it. But in that case, we can't do the automatic reference frame capturing. So then you would have to manually select the reference frame. I think I can briefly show that in a minute. So now we measured these 20 cells before and after ISO. And I'm practically done. So I just stop the experiment here and stop it here, and I will save this file. I'll just put it to the demo data. And this is then the complete file of all the measurements. And if I open a new one, I can briefly show how you can also manually detect the reference frame and see if you have captured the correct one. So now in this frame, you can see the image of the contraction, but we also can look at the plot. And then you can press capture, well, roughly on the correct moment. So if they're beating fast, that is tricky. If they're beating slower, it is easier to do this. So if I look at the video, once you can click it, but as soon as you leave the button, then it's actually the capturing happening. And then you can see in the plot if you actually found the right one. No, clearly I didn't, because you get a very strange shape of the measurements. Now it does seem to be correct, but actually it's flipped, because pixel intensity should go up after diastole, while pixel correlation should go down. So if you have to do it manually, it is a bit more tricky, especially if they're beating fast. But it is a very small system, and it can be attached to any microscope. And with just a laptop, you can have an idea of your contraction of the measurements. So this will be a way to completely manually do it if you don't have MultiCell light or MultiCell at all. Right, let me also show the automatic finding of the locations where you can measure. Okay, yeah, so we're in a region that is more or less contracting. I press start here, and I can show you how I can set it up. So for adult cardiomyocytes, we had a method to find them automatically. For stem cells, we have one that's still roughly in development, or at least in validation, which is called center of contraction. And you can use that to set it up. So the only thing you can change is the binary threshold, which is not very specific. So what this method does is it finds what is called the divergence, and the divergence means how vectors come together to one point. And if they all come together to one point, the divergence is high. If they all move in one direction, so all vertically or all horizontally, it doesn't add up to the divergence if you calculate that. Other than that, you can set the area of the divergence, but that's more or less it. And I can just show you what it looks like. So if I just press find a measure, it will just start finding locations. Let me switch here to pixel correlation again. So while I'm doing that, it's still looking for regions. So it goes to a new field of view, and it does the calculations of the centers of contraction, and it can find one location to measure, or it can find multiple locations to measure within that field of view.
We got another question from Brian. Sometimes I get crazy baseline drift when measuring human iPSC using manual selection in a non-multi-cell system. What can be done to mitigate the drift?
Yeah, we also sometimes see the drift. We're not completely sure where it originates from. It is related to the reference frame, because you see the baseline also dropping slightly. So it's something in the complete background intensity or the background light changes slightly. So that could either be the solution inside the dish that has some movement that affects the total view. Other than that, I don't have a very good advice, I'm afraid. Yeah, so it found 10 regions. Not all of them. Well, main part of it has nice transients, but well, more in the last part, you can see this one was not a location that I would measure manually. So this center of contraction is still in development, and we try to optimize it. but it could be helpful, yeah, either way. I think it's time to switch to the analysis, unless there are more questions about the acquisition.
Nope, let's move on.
Okay. So for the analysis, we use CytoSolver. And I can just import a file. So I'll pick the one that we just created, and also a couple that have calcium. So you can also see that if you have it in a EBD dish and you load it with calcium, you can also still measure that. Oh wait, this is not calcium. These ones are. So you can either select one file or multiple files, and those will be analyzed by a CytoSolver. So what it does in the case you don't have pacing marks, it will find the transient, or it will find the peaks of the contractions, and based on that, it will find the start of the transient. So in this case, this is the file that we just measured. You can see that we have both pixel intensity and pixel correlation that we both measured. You see the whole file in real time. So you can see the first measurements. These are all from the first round. And then when we did the ISO, you can see that all measurements were done in very a narrow time window. If I just click on one, you can see the trace. And if I want to also see how it detected the start of the transient, I can add that here to add the transient limits. And then the yellow line indicates where it starts with the transient. So it is a detection of the peak. So if the contraction is very small, or if there's a lot of noise on your data, this detection can be off sometimes. So if you have data, or you get strange data, or the transients are difficult to be analyzed, it's always good to check if the peak finding was correct. Based on this peak finding, we also get the beat frequency for each segment and the beat frequency variance. And I can just scroll through all the samples. You see the segment and the sample number. And sometimes it rejects a trace. Well, at the end, this one is rejected because there's hardly any number of data in here. But you can see also for irregular data, it finds these starts of the transients quite well. So you can see also in the percentage here, it indicates the number of transients that we're not able to analyze. So 18%, that's usually just one at the beginning, or at the end, it just misses some data. But if it's higher than 30%, it will also be highlighted by yellow. And if it's higher than 50%, it will also be highlighted in red. So you have an idea of, okay, I need to check that trace or that segment and see what happened. Why is that measurement not that great? And then you see after sample 20, I go back to sample 1 because I did repeated measurements. So this again is sample 1, it's segment 22, so measurement 22, but it is sample 1 again. And I can just scroll through that and see why things have been rejected, or if it was detected well. So this data is all collected pretty well. There I don't see any yellow or red marks. For each of these transients, we do analysis on them. These are the same as we do for adult cardiomyocytes. So we fit this transient. So we try to fit the peak, so we can find the time to peak. And we also fit the relaxation part of the curve by a single and a double exponential fit. So you can also get tau, but we also have a polynomial fit that will just give you the time to baselines. So from 10, 20, 30, 40, 50 to 90% of the baseline. And it does that for each trace separately. And all that information can be found in the data tables. So if I scroll down, I'll try to do it slow because there's a lot of variables that are given, but for iPSCs, a couple of them do not make that much sense anymore. They do make sense for sarcomere length measurements or edge length measurements, but not for pixel intensity or pixel correlation. But the ones that do are relevant are time to peak, for example, and you have all the time to peaks in between. So from the very start till the very end, time to peak 90, and also the return, the time to baseline. So the relaxation is also fitted. You get that also for the different time points. If we look at the segment data, we get the beat frequency analysis. So again, you see each sample. So we have 1 to 20, and you have the repeated measurements. So that indicated if I had multiple runs. So you can see here, I had 20 samples measured, and then I switched back to number 1 of the second run. And if I scroll through that quickly, you can see here it's roughly 2 or 2.1 hertz with some exceptions. And if I add ISO, it increases to 2.5 hertz. So a slight increase in beat frequency. If you want to look more at the variance, or if you're interested in if the beat frequency is I'd say it very strict or the same all the time, or if there's a lot of variance in the beats, you can look at the beat frequency variance. And we also have the beat frequency standard deviation to give more of an idea of beat frequency, well, changes. It is interesting to note that it is relevant to look at the number of beats. So for example, if you have very slow contracting regions, you might only have 2 or 3 beats within your measurement. Then the reliability of beat frequency variance is lower than if you have 10 or 20 beats that we could have analyzed for variance. So if you're really interested in looking at beat frequency and beat frequency changes or variance changes due to compounds, then yeah, it is advisable to measure for long enough times that you have really a good number of beats that could be analyzed.
Another question from Brian. Can CytoSolver give raw data points and or create composite traces?
Yes, it can. So if I look at the segment average, then we get it per segment. We can also have that for the complete trace. And I'll show in a bit how you can also export all that information. And then you can either choose if you want to have the raw data in there as well, or only the variables that we analyzed. So if I want to, I just quickly want to show you how it looks like if you do calcium measurements as well. So in this case, I have both rescue calcium and pixel correlation. And you see here that, so then in calcium, yeah, the calcium trace looks similar to what it does in adult cardiomyocytes. iPSCs are relatively easy to load with Fura. So we use, in our hands, we use a 0.5 micromolar of Fura2 to load the cells for only 15 minutes at 37 degrees. And then we wash them twice, and then that's it. So that's relatively easy. Unfortunately, I couldn't show that today, but that is definitely possible. It's also possible to combine it with a FluoVolt experiment, but then you have to change the calcium dye as well. But there's an application note on that for those interested. If I want to just focus on the measurement that we did today, I can also annotate that information. So now we just have run 1, run 2 in the export, but we can also make it more clear that run 1 was thyroid and run 2 is ISO. And for that, we have to first set up an experiment design, which can be helpful. I'll start a new one, demo 2. I'll just call it demo 2. And we start with one single node in this, which is usually the construct or the species or anything related to your experiment. And then depending on how your experiment is going to look like, you can have this either for control and maybe, oh, sorry, control should be placed here. The genotypes. So if you have different cell lines that you want to compare, you can have control cell line, you can have a mutant cell line. And that way I can build up my experiment design. So now I just have, well, today I have control data. So I can just add what we did to the conditions. So we did thyroid and we did ISO. And that way you can build up your whole tree. And if you have that tree built up, you can annotate the data with it. So if you have a file that is only control data, you can annotate it as control. And if I have run 1 is thyroid, I can annotate it like that. And you can make it as complicated as you like. So you can go multiple levels down if you have different pacing frequencies that you want to use, or incubation times, or any design you might have, or you have a large amount of compounds or a large number of different cell lines. You can just add them to the experiment design and you can annotate your data with it. So if I create this this design, then start to annotate with that information. So this is my file, and that file contains 2 runs. I have one run which was the thyroid, and a second one that was isoproterenol. So first I will annotate the first run, which is an iPSC. I have controls today. No, the first one was thyroid. I annotate. So it tells me already I've partially annotated the file. Then if I switch to the second run, I only have to switch this last one to ISO, annotate, and then press OK. And then all that information is now added to that ZPG file or to the data when I export it. So if you export, there's a couple of options. You can just look at all the data of the average per segment. So you get one variable per measurement, or not one variable, but one readout per variable. Or you can look at all the individual tranches if you're really interested in the variability within one region. In general, we look at the average transients, especially for calcium measurements, that in the average, it improves the signal to noise ratio of those measurements, and thus also the analysis. And we can export it to TSV file, Excel, or both. And the option indeed to export the raw data. So if you want to reconstruct the traces, you can use the raw data for that. But we also created a tool to look at this data, so I'll show that shortly. So for that tool, it has to be at least exported to TSV file, but for your own analysis, you might want to use Excel. I'll just press export.
While that's exporting, another question came in about the experimental conditions. Francisca asks, how much calcium do you use in the thyroids?
Oh, I had written that down. I think we have 1 millimolars of calcium, if I remember correctly. I can't see the paper. No, we usually have, I think, 1 or 1.2 millimolars. But I can look that up. So for visualization, we have the DataVis tool. which is opened here in the browser. And with that, I can import the file that we just exported from CytoSolver. So that's this file from today, this time. I could just take that, give it a second. And as we had both pixel intensity and pixel correlation, it will show you the information of both. And I can show them side by side as well. And the first, what we see is the number of data points. So that should be roughly 40. And on the y-axis is the parameter of interest. Now it shows baseline, which is not a very interesting parameter to look at, but we can switch that to time to peak, which you can still see, but you have still no idea what you're looking at, what was the condition. If I change this one to conditions, you can see the ISO and the tyrode being colored. It doesn't appear that the time to peak has changed that much with ISO. Looking from this, we can also look at the time to baseline, 50. So all these variables that we have from CytoSolver also now in this part. So you can also look at time to baseline. Again, there might be a small change in time to baseline. We can also look at the summarized data to see if it looks slightly different. So there's a slight, so on the left is ISO, on the right is tyrode. That's pixel intensity and pixel correlation. So they should roughly show you the same thing. So you see a small decrease in the time to baseline with ISO. What we can also create is a new parameter. So for example, if you want to look at peak width, if that has changed, that's also something we can create here. So if we want to have the peak width 50, so that's 50% of the peak width 50. We can calculate that based on the time to peak and the time to baseline 50. And then by taking time to baseline minus time to peak, we calculate the peak width. So below you can see the result of that calculation. Wait. I create that variable, and then it will also show up here. And we again color it to condition. And you see roughly the same effect. So slight change with ISO, but not a drastic change. We can also look at the average traces per condition. That is also interesting to look at. So you see it for pixel intensity and pixel correlation. So pixel intensity, it all goes up. You see there is a change in the peak height and hardly a change in the kinetics. What we did see was a change in beat frequency as well. So the response to ISO is there, but it's not dramatic, or it's not, you don't speed up the contraction that much so far, at least not in this these cell types. I think that was what I was wanting to show. Not sure if I missed anything. Already in for an hour, so that was roughly what we scheduled it for. So
great, thank you. I mean, so does anyone have any other additional questions regarding either the acquisition, the analysis, or the data interpretation? Oh, we got one. But you wouldn't necessarily interpret the change in peak height to ISO. So Andrew's asking, even though you see here that there is a change in the peak height, you wouldn't necessarily interpret that, or would you?
Well, it's tricky because it is repeated measurements. So I think there is information in there because you measure the same location twice. I think there is information there so that you activated maybe more cells to the contraction. It can be both. So either you have more cells contracting in your region because of ISO, or the cells that are contracting more. And you can't really pull those two things apart. So there's information there, but it's not as straightforward as you might like.
All right. I don't see any other questions coming in. Thanks again to Emmy for the demo, as well as to the VU for providing the cells. And thanks to you for coming and joining us. Again, we'll be uploading this presentation along with the application notes to our website. I'll let you know when they're available. If you're interested in any of the products that we showcased here today, or you come up with any questions later, please let us know. You can visit our contact page on our website to submit questions or inquiries, or you can simply reply to any of us directly via email. I know that my colleagues Amos, Johnny, and Adam have contacted many of you already, as have I. And if you're unsure of how to reach us, feel free to use our general email, info@ionoptics.com. Thanks again, and have a great rest of your week.